No problem. I hope you can hear me, right? Yeah. Okay. 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 So the title is extract. Extract means your like most of the research projects I have seen at my time and even today are plant extracts. So the plant extracts using a short-term acute toxicity study. So actually, Dr. Jagadish shared some of the thesis, which has actually like a nice pharmacodynamic study results. But on the other side, they end up with talking about the toxicity by mentioning the short-term acute toxicity, which may not be accurate for all the research projects if you want to talk both pharmacodynamic and toxicity. So because like you need to establish a therapeutic window to discuss about like the how safe it is when you want to take this research to the next level, like for example like clinical situation. So in any studies like just before going to the talk, I need to mention this. This is a disclaimer. All the words I say or any conclusions I made, like that is all my personal. Like, of course, this is Dr. Jagdish and mine, or nothing to do with his former agency or my current workplace. So please don't take this as a reference for any claiming of any data. So the question which we are going to discuss here, does the dose matter? The answer is simple. Dose does matter always, like in any study, because like that dictates your pharmacodynamic effect or pharmacokinetic, like toxic effects. So these are the classic examples you might have seen in your textbook, right? Like the dose response, and when they make it to a cumulative frequency response, like you see this. One slide back. Okay, I can do that. So you see this nice sigmoidal curve. So I think in any research, this is what you do. You establish a dose relationship. So that is a, by definition, it's a quantitative relationship between the test compounds, either its concentration or the dose, if it is an animal study, or its exposure, like if you consider a pharmacokinetic data like AUC or PK, like Cmax values, to a pharmacodynamic or toxicological effect. This, I would say, this is the basic unit of any research, a relationship with the response for the concentration, like to the concentration. So the sigmoid curves could be many things, like you if you are using within the therapeutic range, that would be a, like you can establish an effective dose, pharmacodynamic activity. So if you do a toxic dose, so that would end up with a toxic dose, like you can establish like a toxic dose. Between these two sigmoidal curves, like if it is wider, that gives a higher therapeutic index. That means you can play the doses like little easier. But like if it is a bit narrow, then it could be end up with like your pharmacodynamic activity could overlap with the toxic effects. So that is very important to when you take this your research to the next level, like if you want to use in the clinical, because like all the data we generate in the non-clinical studies that are actually help to design a dose for the human, like a human trial or human use. So on the other side, this lethal dose, which generally like as Dr. Jagadish mentioned to me, some of the research proposals to talk about like the toxic level. But most of the lethal, like in fact, like all of the lethal dose studies, they are end up with conducting with a single dose, means high doses they give. But this is not a representative of number for establishing a therapeutic window. because we use higher doses. But like all the your research, like for example, like your plant extract or your newly synthesized drug, they you want to use for a diabetic patient. So that need like a lifetime treatment. So the single dose may not give a right answer, like right result to talk about the toxic effects of the drug. So it is important to establish like this kind of data to make a judgments. So to just go to further into this, like this dose responses, you see the in vitro studies, like usually you do the pharmacodynamic studies, right? So that is for the mechanism studies. So you have a drug or you have a plant extract. Sometimes the plant extracts, you may not be able to do all the in vitro studies because it will end up with a lot of chemicals like molecules. So it may be difficult to understand, but at the same time, like it is also possible to do any standard assays, like if they are available. So a cell-based assay or enzyme activity study or any um, like the receptor interaction studies, like dynamic studies, that would establish a concentration that can inhibit like a 50% of the drug activity, like 50% of the activity actually. So that gives a EC50, effective concentration 50, and the dose levels usually, like if it is a single molecule, you would end up with the picomolar or nanomolar or micromolar concentration. Usually that type of graph would come here actually, another sigmoidal curve. So, and when you have the mechanism studies and like your compound is so much active on the receptor, then you will have you would know like 50% of the concentration, 50% of the effect, what concentration is needed. And the next level is the in vivo study. You need to establish a proof of concept, like that your mechanism, like whatever you have seen in the cell-based assays, or translating into the disease models. Again, like this is in vivo animal model study. So in the proof of concept studies, like if at all, like you may based on the availability of the right animal model for the disease you are aiming, you may end up with conducting the studies in the normal animals that are healthy animals, and also disease models that are like either chemically induced or transgenic animals. So again, like once you get this effective concentration, like ED50, effective dose, that is like 50, you can calculate, and this will be because like you are dosing the animal with the mg per kg, either like per day or per week, depending on the type of drug, and you would end up with the ED50 value. Thank you. So, and then the other curves are like the toxicology related, the toxic effective dose. These are based on the safety studies. So you will be having a regulatory toxicology studies. They are available like the guidelines from OECD or ICH. You can go through if at all like your research is demanding you to conduct any toxicology studies. You would establish like you generally conduct like a dose response finding study in which like you may end up with finding like a maximum tolerable dose, like that is like a dose that can have like little bit of toxicity as well as like mostly like the toxic dose. And then sometimes like depending on your type of compound, like you will have some limitations to have the higher dose that is like maximum feasible dose. So again, like you establish like the no observed adverse effect levels are low L's, and then you would end up with if you use this lethal dose, maybe you will even end up with the TD50 values. And again, these are like same as like milligrams per kg per day. So these are the important aspects like in your research when you conduct, like when you establish any preclinical data to support your future experiments. So all these doses are important for you to make a call for human dose. So other aspects of the study. So the study design, it has to be clearly defined objective. And like as Dr. Jagdish mentioned before, like how you write your hypothesis, maybe the experimental part will be discussed a little later. The endpoints and you need to have an appropriate controls if you are using any animal model like as a reference to establish that the model is working, and also you can have a comparison. So compound characteristics like the stability of the compound. So you cannot say like I have a compound sitting in my refrigerator from the last 2 or 20 years, but it's still the same. No, that is not true. So you need to have a right compound to test, and the route of administration usually the same as the clinically indicated route of administration, and there could be an exceptional depending on the formulations or any chemical issues. And the environmental and experimental conditions, these are also important. Like you cannot keep your animal on the street and conduct your study, because like the stress itself can cause so many different brings a lot of variations to the study. So you need to have that, like the conditions appropriate, like the temperature or like the light cycles, humidity, and like the person like in and out, like those are the things you might not say in the academic research, but once you go to industry, particularly the pharmacology students, or even like anybody who are in the pharmacy, you will experience this, like the need for these kind of situations to conduct a clinical study, like a preclinical study. So, and also the model should be validated, and the like one time interval or multiple times, or like the main study versus like your satellite group if you want to include any pharmacokinetic groups. So there biomarkers, if you have any plan for using a biomarker as a endpoint, so that should be appropriately validated. And data analysis and interpretation, these are all will be covered in maybe in the other sessions of this workshop. The main important point is the dose selection. So the selecting dose should be based on the preclinical data to balance efficacy and safety within the therapeutic window. As I said, like you should not be getting a toxic dose, and you still, because like your endpoint is only pharmacodynamic activity, you consider that dose is safe, but like that may not be true. So you need to establish that therapeutic window. So to start with, you can have a literature review, like usually, like if it is a plant extract, you may have already some data. And if it is a chemical molecule, like you're synthesizing has like some other molecules already approved or in the market, there may be some literature that can guide you. And like even some chemical structures from which like your drug is derived, like as an analog to the parent drug, like the main molecule. So that might also have a therapeutic window. So there will be some data available. So that could be your starting point. So there will be in vitro data. If you are conducting any in vitro data, like cell-based assays, so that also tells, because like your compound has more potent, that means like you have more chances of having more toxicity as well, like because potency sometimes like on the target maybe same, like maybe similar folds like you see in the toxic side of it also. So it is important, like if your in vitro data can also teach something, like, and also people use, like when in the industry, like you need to have the pharmacokinetic data to support your receptor, like for example, like a cell-based assay, and then what is the percentage of that exposure in the animal in interpreting the pharmacodynamic activity in terms of, again, the dose relationship. So the safety factors, sometimes you have to use depending on the species or route of administrations, etc. So, and then you would select a range of doses to determine the optimal dose response relationship, because like you cannot just throw some doses, like you need to have some thinking, some work on the literature, like the existing data, and then plan some doses and justify the doses. Like you all need to end up with a study, like you are sacrificing a life for your research, right? Like animal, like you are sacrificing an animal, like that that needs to be justified. You cannot just kill. We don't have a right to do that. Like you need to justify why you are using. So that's where like you need to do some good research before you selecting the doses. So there again, like as I said, like the maximum tolerable dose. So this is the highest dose that can produce acceptable toxicity without causing severe adverse effects or mortality. The key considerations, like you in the studies, like you can see, particularly in the toxicology studies or dose range finding studies, you would end up with clinical signs of toxicity. Because like in the toxicology studies, like you would measure the like the behavioral aspects and also your laboratory, like clinical pathology, like clinical chemistry, and the pathology, histopathology data. So that all can together tell you like what is the dose. And this sometimes like there is a limit dose, like sometimes like these plant extracts may not be toxic, so you would end up with the limit dose. So the dose spacing, like you don't do one single dose, like you have to have, like to get that curve, you need to have multiple doses. So for that, like you have to space, you have to select a dose spacing. So that could be high dose, maybe close to the MTD, and the low dose typically, like when you establish a NOEL, that would be your low dose. You will adjust the NOEL middle dose in between. Like I'll give more details about that. So another thing is the maximum feasible dose. As I said, like this is upper limit is dose is dictated not by the biological or toxic dose, toxic range, but like a practical feasibility. Your compound is not soluble. For example, like the volume of like here, there is a limit for dose volume, but like your compound is not soluble. So you may end up with maybe 2 times a day. So that kind of feasibility you have to address. If you can't address, that would be your limitation to conduct the study at that dose. So uh, there are other factors like physicochemical properties, formulation challenges, administration challenges, for example, like viscous solutions or the animal tolerability. These are all the factors that would dictate your dose, other than the toxicity that you would see. So uh, in the dose range selection, you would use multiple approaches. That could be an empirical approach. There could be off-log or threefold scaling. For example, like 3, 1, 3, 10, 30, and 300, something like that. Or scientific or mechanistic approach, that is like a pharmacokinetic data. That is like if you have like available data, pharmacokinetic data, that would be helpful to decide your dosing. And the allometric scaling, like you again, like you can predict based on some of the existing data, then other aspects like if you are species correction or human corrections, like additional factors, you can do the PKPD modeling and end up with the predicted human dose or something. So those are the ways, and also the like there are some guidelines that has recommendations why you can use. All these I'm talking about is may not be applicable to the research, but you would see this. So you have to start thinking, like you may not use that in your current research, but like you need to understand why the dose selection is so important to support your research or make that research to take it to the next level. So you need to understand, maybe if you don't do that in your research, but still you need to go back and look at all the available information and then understand, so that like that would be helpful for your success in the interviews as well as your current or future research. So like as I said, like the other doses, like I mentioned, are all about the pharmacodynamic studies. So, but for the toxicology studies, you would end up with no observed adverse effect level. Like you need to understand, you need to establish a dose that won't show any toxicity. So that means up to that level you can go. So there are some guidelines you would see that are available, like how to conduct those studies. ICHM 3, for example, and again, like the dose selections or low dose should be around like the. I think if I press little longer, it's dropping out, I believe. Oh, okay. Okay, okay. So the study duration is depending on the requirements, like there are guidelines that would say either like to support any toxic dose, how many days you need to work, and the TD50 values are like 50% of the animals that are showing toxicity. So typically based on the acute and chronic exposures, so you would end up with some data in these toxicology studies like organ toxicity. So that would guide you how much you can use. So this is all helpful. Like why we're doing preclinical, like to support your clinical trial. Like so, don't underestimate the potential of your research proposal. Like, but like at the same time, don't do a right thinking like and selection of the doses so that your even if it is a small data you generate that might be useful to establish a drug in the future. So when you conduct a study, use all the standards that you need to use to establish to generate a pure useful data. So we should not be, somebody should not be thinking that like, oh, this is coming from this place, this data is like it's not validated. So I actually like that is not true. You can actually do some research and existing data and plan your research in a way that can actually generate a validated and reproducible data. So then it start believing like all our research. So that to bring to that confidence to the everybody, like I think whoever getting into the research field, so they have to understand the standards, and like Dr. Jagadish is giving you a wonderful training today and tomorrow. Make use of it, like learn the principles and make use of it. So so once you establish the toxicity and the pharmacokinetic data, you will establish with the safety margin for the animal studies. So incorporate a safety margin to ensure dose selections are relevant to clinical application, as I said. So there is an example here, like if you sometimes like you expected to have 10x, like 10 times the safety margin to anticipated human exposure, that might be like a best bet that you wouldn't expect to see any risk in the humans. So you can actually refine your doses based on your experience and like make it right dose at the end of the clinical study. So as mentioned, like there are acute toxicity studies data that are used to talk about like the research proposals, like, oh, I have the LD50 of this much, so this drug is safe. Although it is not true, as I mentioned, it's better to use the toxicity. But I just wanted to talk about like the existing guidelines that will talk about like how these studies are conducted. First of all, like the OECD 401, this is a multiple doses. Probably at the same time they do all the dose levels and wait for the animal to suffer and die. And then 50% of the animal, wherever, like whichever the dose they get. But this is not a humanized way of doing a study. So that's where like the new, like we are evolving, like we are learning, and we are also like generating a lot of data. So based on that, the new guidelines came. So that are like have a different purpose actually. So there are like OECD 420, 423, and 425. They have different purposes and in establishing the lethal dose. So why we need multiple guidelines? Like as I said, like the ethical consideration and implementation of 3 Rs. So that are like Rs are like representing like the replacement and reduction and refinement. So wherever possible, you have to reduce use of the animals. Like as I said, like I'm stressing again, we are sacrificing a life for our research. So be responsible for that. So generate the data that is useful, not like your thesis or you a piece of paper that is like satisfy your requirement to get your degree. You can do that, like you had to do this research anyway, but do it with responsibility. That way, like you generate a data that is valid and useful, and it can take it to the next level. And the regulator, like as I said, like these three guidelines, 420, this is actually used to screen the large number of chemicals for quick understanding of the toxic label. And whereas 423, this is used to classifying chemicals under the regulatory framework, like the GHS classification. You conduct this study, or there are like I'll discuss more details of that. But you end up with classifying the chemicals. That way, like you generate like that can indicate that okay, this drug is this much potent in producing the toxicity. So 425 is a more precise value. All these two are giving like a range, but this is you can use the additional mathematical application like calculations. There are softwares also available, like free softwares, I believe, so that you can use to generate this data. So this is a comparison table, like the dose levels are like fixed for 420 and 423. This is like more of dynamic, although there are reference doses you can start with. The initial testing with low dose, and you can do it like if you have some data available, like you can start with the middle dose also. And this one is like more of range finding. This is for the classification of the chemicals. This is for the exact estimation of your LD50. So the animal use is less compared to the 420. So there is more responsible way of conducting this lethal dose toxicity studies. So this is the 420. You see in the OECD guidelines this schematic diagram. So there are like 5 doses here. You would start with your first dose, one animal, not all 5 animals together. So then if there is a death, then you would classify this as a GHS1. So if there is only toxicity, still it will be classified as 5. But like if there is no death or toxicity, then you would go to the next level. So you will continue this until you establish like this. This actually you can use a single sex in the group, or like the female is more sensitive based on the data. So that female animals are generally used for these studies, one animal at a time. And the dose cause evidence of toxicity or not more than one animal death, you can continue. And like 24 hours you have to give the window, because animal, like you might not see all the observation within like three hours or 3 to like less than 2 hours. So you have to give the time. And like the observation period, like as previous 420 401, like similar to that, like you have to have the observation, because like there will maybe delayed toxicities. So you need to have that time period for this. So the same way, like the doses you can change. Like if at all, like if you start like with the 300, assuming like you have some data already. So then you start with, when again like you end up with some death, then you will actually scale down. But again, like you will be using initially, this is there's the 2 parts. This is the sighting study. So once you have this one single animal or a couple of animals data, then you go to the main study, in which you would use 5 animals. And again, like you would end up with your classification is different here. If it is 8 or more 2 deaths, then you would end up with the same dose. But if this less than 1 animal, then you would classify differently. And if there is no toxicity, you can go further. And again, like you can scale up or down depending on where you start. So this is for the 300 and 2000. And all these guidelines can go up to 5000 dose if required, but like 2000 is usually a limit dose. So this is another guidance. This is the stepwise. And the first step, you would conduct like the 3 animal studies. And then like based on the data, you would end up with like categorizing them in the GHS classification. And then like if you have like less severity, then you would conduct like another 3 animals. So that way you actually reduce the animal use, and also you generate the right data. So that's the same way as I mentioned, like 420. So this one, like you can change the doses. You can start any doses that, but like you can either scale up or scale down. So this is another guidelines. Maybe this is the one like maybe researchers use more often to talk about the LD50, because LD50 you can exact number, or if you conduct like the proper experiment, you would end up with getting the right data. So this is also like has like 2 parts. One is the limit test, like there will be you will be using, like if at all like your plant extract end up with no known toxicities. So then you would start with a limit dose, that is 200 mg and 1 animal. And if the information indicate that the test compound is less likely to be toxic, then you would establish 200 as the dose. But like if the animal like if there is a death, then you would conduct the main study. If the animal survives, uh, you would conduct another 4 animals. Like if you end up with more than 8 or more than 3 animals, you will move to the main study. If the survival is seen like in 3 animals out of 5, then like you can actually conclude up to 300 mg is the safe dose to proceed. So in the main test, like there are some reference doses, like you can use, as I said, like all the 3 guidelines, you can go up to 500 mg per kg. But like the dose levels you use is like with a half log or 3.2 progression, which is, as I said, like 10, 20 or any of these doses, 10, 30, 3, 10, 1, 3, 10, 30, something like that, and they are similar to this one. So if you don't know much information, maybe you can start with 175, but usually this limit test would guide you to select the dose. And like you can step up or step down depending on the results. And there are stopping criteria to conclude the LD50 value. But like that is not the end, like you interpret that using this softwares, or you can use actually formulas that are all available in the guidelines. So you can use and establish the LD50. So this is the importance and the need for the dose selection. Just to summarize, your high dose in the pharmacodynamic studies are typically chosen to maximize the objective pharmacodynamic effect without introducing any severe toxicity or systemic adverse reaction. So you may use either MTD or LD50 values. So for MTD, once you get from your dose response study, the MTD dose as your high dose. So for compounds with the plateaus, this is not that simple. Like when you have a combination of data between like your toxicity or pharmacodynamic study along with your PK, you would see some saturation PK parameters. So where you need not to go to the high dose, because like the no further exposure, like for example, like you're using 10, 30 and 100, and 30 and 100 have the same PK data, then 100 may not be a right dose. Because you're unnecessarily adding like more drug to the body. So you can end up with 30 as your dose, because that itself has similar pharmacokinetics. So like although I'm talking about like the 2D structure, but in the industry or in your real research, you would end up with maybe 8D, like 8 directional data you had to collect to make a call. So So that is important when you have a PKPD, pharmacokinetic data is available. And the lethal dose, as I said, like there is a general rule, like there are like how many folds less. If you have to rely on the LD50 value, there is a rule that this is the dose you need to establish. Like you can't say that like, oh, or like my lethal dose is 10 mg, but I'm doing my study at like 8 mg or 10 mg, because like that can either kill the animal or end up with the other the toxicity. But like, because you're not measuring the toxicity and you're only concentrating on the pharmacodynamic effect, that doesn't mean like your data is valid to use in the human situation. Because like if I don't take a drug that can produce a pharmacodynamic effect, also toxicity. I don't want, like I need a drug that can only target like what the disease I have, not the toxicity. I don't buy a drug for killing myself, right? So you need to, like being a researcher, you need to have that different, like when you talk about like plant extract so and so, or like this new synthetic compound has this pharmacodynamic activity, that means it is safe to use at that dose when you extrapolate to human. And you have to establish the therapeutic window that it has this many fold safe, even if you use in the clinical situation. So that's how you establish the data. And the mid dose selection is like you can use this dose response curves, and you either like empirical spacing, like as I mentioned before, and the mechanistic based on the PKPD, you can do that. And the low dose is again like the same, but like with the different folds. And if it is close to the NOEL, that would be good to have that right dose selections for your studies. So again, like one study may not tell you everything. Like when you see a single dose study, you end up with a dose range differently than when you conduct that same compound for 28-day or 90-day. So there you would end up with a maybe more toxic at lower doses. So you have to refine, like depending on the clinical indication, again, like you have to change your dose selection. So you have to be flexible enough to propose what could be the uh, 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 condition, like if you use, because like if you are talking about toxicity only based on the lethal dose, that may not be accurate if you propose this drug for a diabetic treatment who needs a lifelong treatment. So you need to establish that safety with reasonably like the duration of the study. Your clinical duration of study duration should be supportive to your clinical study. So for example, like this is like just to summarize everything, you end up with 300 mg. This is a single dose study. And like in a dose response, you end up with 50. That's the maximum. Like anything above 50, you're ending up with a toxicity. And then like yeah, the lowest no observed adverse reaction is at 5 mg, and your PKPD modeling shows based on the EC50 value receptors interactions like the 20 mg. So I mean like actual concentration at 20 mg. So you might select high dose close to MTD, that is like 40 to 50 mg, and the mid dose is 10 to 15 between these doses, and the low dose is around 5 mg per kg. So this way it ensures that like the pharmacodynamics are observed, like separated from the safety concerns. So this is all about the dose selections in the preclinical studies in your research projects. Uh, just like I'm open for any questions from you.
Raise the hand. So
Room is open for questions. Does anybody have any questions? Please raise your hand.
Please don't raise your hand if you don't want to ask questions. Did I say it correctly? Yeah. Okay. Negative, negative. See, I talk about like 30, 40 minutes, right? I'm expecting at least 4 or 5 questions.
Good afternoon, sir. I'm Kajal.
Yeah.
So, like while we were doing preclinical studies, like during our project, so we were confused about the dosings, how to decide whether if it is like toxic or not, because we don't have any another measures or any other methods like we can identify whether it is showing some toxic symptoms or not, like animal. So we started with like a very small dose, like 10 mg, 20 mg, 30 mg, and we don't know where to stop. Way to stop, we don't know. So, like, are there any physical methods, like by visualizing the animal, can we identify that it is toxic or not?
So you that's why like you might conduct it is necessary to have a DRF data like the dose response relationship. Like if you establish that data and incorporate toxicology endpoints, that is mainly like you wouldn't bother about like whether you're getting a pharmacodynamic data from that study, but like at the same time, like if you use that for like a behavioral effects, for example, or like like all the Irwin tests, like if you are using mouse, you would see what kind of toxicity information you can get. You may not get the cardiovascular, but like you can see the animal like behaving like a respiratory deep respiration or something that can indicate like those clinical observations of the study might give you some data for selecting like a dose that like then like if you end up with like that you can consider it as like a potential MTD, maximum tolerable dose, and from there you can scale it down to 1/10 of it or something like, and then fix some dose ranges, and then from there you can go, and maybe Dr. Jagdish can add additional points to that.
Potency and efficacy of the drug, then you have to play around with the toxicity. So
So for rat, one mouse, we have injected a dose of 50 mg. Let us say vanillic acid, and at 50 mg, it started to show some cardiovascular depressions or respiratory depression. Then should I conclude that 50 mg is a toxic dose, or I can?
therapeutic part we are talking about.
Yeah,
Because
therapeutic
you are
part.
able to get some response at 50 mg. What happens if you do with 100 mg? Does it stabilize at the same response will be there, or does it increase, as I said, dose response? So does it show more efficacious points or not? Or does it go to stabilize? Because usually it goes like this way, then gets a stable point. If you are 50 milligram,
After this,
you are having a
is going
stable
to
one.
come to you, okay? So be prepared for the second question also. No, I'm just encouraging people
If
to
it
ask
is
questions.
if it reaches a plateau with your response, then there is no point in adding anything more. That itself is a maximum dose. For example, decreasing the blood pressure. It already decreased about say for 50% response already decreased. That's fine. You are able to demonstrate the compound is hypotensive. It has decreased the blood pressure. So you don't increase much more. If you increase further twofold or fivefold, if it doesn't show further response, that is your maximum dose. So therapeutic point, you're going for it. But if you don't know the therapeutic aspect of the compound, then you go for the toxicological point. So because you don't know, because some of the plant extracts already some literature, somebody has published the work. They've already demonstrated that particular dose is effective. Then you don't have to do any toxicology. Skip this part. So 1 month or 2 months you can save it. But if you don't know from the literature, nobody has done the work. If you are doing in the first year ideas, or somebody synthesize some any pharmacochemistry students who are synthesized some new compound, which you don't know anything about it. We told about in vitro. So in vitro, if you're able to demonstrate an activity, then if in vivo already demonstrate activity, then you know what is the response. But if you want to take a risk, no, I don't want to take a risk because in vitro it has to be done first. Then in vivo you go for second step. For in vivo you are scared to give higher dose, then you go for the toxicology one. Single dose, you said that again go for a second single dose. Then you establish that toxicity. Okay, for new chemical only. In that case, for toxicity usually for plant extracts, more or less about 80% of the cases we know that what is the toxicity of the compound. But if you're using food, it is more active than the regular formulation. Anybody is doing pharmacochemistry, synthetic compounds, having some question? NCE, new chemical entities. No chemistry to students.
Still anyone? So one question I have, sir. You want?
No.
Sir, regarding OECD 423, what you said, we need to go with 3 animals, right? So the duration of study should be either 28 days for acute and 90, will it be the same, or
No, this is
It
actually
is-
single dose study, right? Like
Right.
all the LD50.
so
And like the observation period can be up to 14 days.
14 days, just
Yeah.
one single dose, and we need to observe it for
Yeah.
14 days.
Like depending on the like when you see the onset of the toxicity, you may not see the animal suffering or death within like 24 hours. Sometimes it could happen
Exactly.
like after
Delayed,
like
delayed
5 days.
onset, delayed toxicity.
Yeah, so you have to space the doses just to be humanized, behaving more like a human. Like you need to respect the animal sacrifice there. So your period should be enough spacing to record all the observations. Sometimes it may not be, but like even like if you see the death after like 7 days or like even 14th day, you have to consider that is the lethal dose. Like if you have like two animals supposed to or three animals supposed to have that toxic effects, then only the third animal it showed up like on the 14th day, you still go back to consider that as the dose for that.
Okay.
Yeah.
So, out of curiosity, sir, if any death happened, so we any histopathological studies we need to go further to check whether the reason for the death and or what to study that also.
That generally is important in the regular repeat dose toxicity studies.
Okay.
That like maybe lethal dose, it's like observations are the main thing. Histopathology, like you don't, because that is a single dose, like a toxic dose actually. So that doesn't explain. Like usually, like you need to have the histopathology data. Like when you have a organ, like maximum tolerable dose, for example, that gives you the data. Like sometimes, like you can't separate them completely, like the safe dose and the risk benefit ratio. So some doses have excellent like pharmacodynamic effects that are required, but on the other side it might end up with increasing the liver function, like liver cause like mild hepatotoxicity. So those things you can actually observe in the clinical situation. So you keep that as a potential adverse reactions in the human. So then you that suggests like that histopathology data that you generate in your regular toxicity studies can help guide you what are the potential monitoring things you have to recommend to the clinician to check. So sometimes that may be more strong in human because of the species review, or maybe less. So that's how you play. So you can't completely separate the doses, but at the same time, if you have end up with like functional data that shows the liver function is changing, and also you have the histopathology data, or if the histopathology and like microscopic or macroscopic data, if you put together, that could be a severe toxicity. In those cases, generally the study designs are, you see the reversibility of it. So if there is no reversible, like it is causing a reversible damage, sometimes it is still you can recommend the dose for the clinical situation, but if sometimes like it is better to understand why it is causing. Is it like really anything like you can do by reducing the dose level. So it's like, as I said, like the presentation here is 2D, two direction. Like in the real situation, you end up with like 8 direction, multiple direction data you would be collecting and making a judgment call.
So one last question from my side, sir. What do you suggest finally, whether to go for with fixed dose, repeated dose for 28 days or fixed dose for this 423? What would be suggest? What you
I
advise?
thought I put this answer in my
Yes,
presentation.
yes.
But like, see, it depends on what is your purpose.
Yeah.
So if you have like synthesizing, your chemistry colleague synthesizing like 10 compounds and you want to see which is less toxic to start with, so then you would go with the first one. But like you want to like that chemical is something like a paint or something like sometimes like you may have to provide the GHS data, for example. You would go with the second one. But like if you are using for the LD50 as a some kind of like dose selection in your studies, the third one, like I think that more applicable to the clinical situation. I think 425 is more applicable for the research situations, at least like for the drugs.
Thank you, sir.
Good afternoon, sir. How do you envision the integration of artificial intelligence or machine learning in selecting a dose or acute toxicity studies?
So there are like already there are multiple like softwares that are available based on the previous data that is like already databases are available. You can like some companies has established this data, so you can use that data by inputting your chemical structure. But artificial intelligence is add-on to that kind of databases. Like you can even ask like what kind of structure can give you less toxicity by that can compare with your subgroups and like the overall skeletal of the chemical structure. So artificial, like it depends, like you're that's a tool. Artificial intelligence, it's another tool to help in making the decision so quicker. Like you need not to waste your time like uh, to get the answer, but it won't replace like artificial intelligence. You have to use what is the purpose of your interest there. Like you want an answer to what kind of subgroup fits well for the receptor interaction. So you have to train the model in a way that like what kind of output you want. So same way, like you can use that for the toxicity. Maybe some kind of chemical structures are known to cause hepatotoxicity. So you can ask a question, hey, I have this data, like this chemical molecule, can it be a hepatotoxic? But for that, you need to train the model with the existing chemicals that are known for causing the hepatotoxicity. So again, like artificial intelligence, generally people use as a like common term for everything. But no, this is another tool to help you. That will help because like more jobs will be created with the artificial intelligence like application. So some people worry that like artificial intelligence can replace your job. No, if you're replaced by a person who learns artificial intelligence. If he knows better application of artificial intelligence, you will be second option for that company. So learn, advance science to the next level and make a quicker decision using the artificial intelligence. That's all. Like it's a tool.
Thank you, sir.
Yeah.
How artificial intelligence helps me in searching for this interactive relationship or once this way we can do it. Any keyboards you can have, put it in, get an answer. You asking the question.
Yes,
The
sure,
first
sir.
presentation. I think this is a good one. It's combined that also in the third presentation. Yes. Actually, it will be searching.
The question is very good.
Good afternoon, sir. Myself, Navnita, I'm from third year.
And nowadays, after the COVID, the 3D technology has raised its wings over all the world, and that can be helped in the means finding out the dose which can be used for a particular system in preclinical trials.
Yes, depends on like what kind of data we have. Again, like see, there is a data, like some compounds will have the structures will have this kind of like potency or causing this toxicity, and you have a requirement for the dose selection. In between, you use this artificial intelligence to help you quickly guide. But like you need to train the models again, like even like you can apply in any way, any place. Like see, you're using your Excel, right? Like for all your calculations, that calculations can be different. But like you use the same formulas, like what input and what output you are asking, like what formula you are using. So that's where this AI will come.
Okay.
Any other questions?
Thank you, sir, for your presentation. So I have a question regarding like completely soluble, and the other thing is insoluble, sir. Whatever solvent we have taken, it is insoluble, sir. In such cases, like how can we proceed with such drug combinations, like
So
for a mice?
see, first of all, you have to try different formulations to improve the solubility. If it is like not possible, then depending on like, is it like supposed to be oral?
Like intraperitoneal, we have gone for IP, sir.
So have you tried DMSO or some hard solvents? Like
Yes,
it's not going.
because the tolerable dose of DMSO, because we couldn't give to the mice, sir. So we have shifted to other like PEG or kind of other things. So
Yeah, that's what. Like you have
but
to.
still it's not soluble, sir. So
Yeah.
that was the
So
critical.
I have seen some of these kind of issues. So the chemical modification is one of the ways, like you generate like a new salt that can dissolve quickly. So you have to collaborate. Like that's where like see, don't think like a chemistry is separate, pharmacology is separate, or clinical separate. You have to collaborate. Like you, when you work together, then you can come up with some new ideas. So prodrugs you can generate or modify the chemical structure in a way that like it can be more soluble.
Because in your presentation you were saying regarding insoluble drug dosage,
Yeah.
that you were saying that twice the dose. So I was
Yeah,
just
that's also possible. Like you make it half and give it twice with a reasonable gap. Sometimes like a morning and evening. So that would help you to get the same dose. Usually, like most of the times we use the per day dosing, but at the same time, like the PK data also important. Like if it is dropping quickly, then you won't achieve the required concentration. So I hope like the 2 time doses would help you get the right concentration to see your pharmacodynamic effects.
Part one is didn't it to be made as suspension. So because only one is interpreted, so you can give orally. Oral giving is always 50% for problem solved. And so in such cases, we use a suspension. There are several solvents are available. Usually, the magic one everybody uses HCMC, HCMC they use it, or they use cytodextron, the one you use it. If you're a chemist, you can always use a salt. That's the correct curcumin. That's soluble.
yes.
So people try like anything. I know in the kitchen people want to do, they put a copper or grandmothers, they should add pepper and use the curcumin. But in the case when you use curcumin along with the pepper for cooking, remember that it increases the absorption much more. But same dose cannot be given because the liver damage, which has decreased the dose. If you're using 1 spoon, you use half spoon when you're using it pepper, or less than half spoon, quarter spoon. The stomach increases its solubility, but as such, if you add curcumin to a boiling one, Heating one, you has a regular amount. Pepper combination is there. That's why to many chemists, what do you make a salt, a pepper, and the footprint together and make a compound? So that is the best one for chemists for doing it. They should do such a way that how we can increase solubility of footprint. Cinnamon, for example, not soluble. Cinnamon is not soluble.
So I was working on berber insights, a natural compound. So it is a very tricky compound. So
Yes.
Any other questions?
Yeah, thank you. Thank you all and wish you good luck and make use of Dr. Jagadish's experience and good luck.
You're eating food and going, no? You're not eating food? Okay. Okay, I'll join in 10 minutes. Let us in case if you are here. Thank you very much. Yeah, I'll be there in 10 minutes. I'll be there. Thank you. Significance of study. Go over to the previous one. Previous two slides. Previous one. This is examples one. Previous 2 or 3 studies when it starts. Okay. The fifth and major arm of the protocol is the significance of the study. You wrote all the protocols and everything. So topic you got it. You got the research question, hypothesis, objectives one. Last part, you have to justify why you are conducting the study. That is significance of the study. For that, how to write it? Many people write review of literature in that section. A lot of students have seen that one. I write it is irrelevant. Only 4 or 5 lines sufficient. You don't have to write methods. Some people write methods. All irrelevant things they put it. Only thing, if you understand what is the meaning of this one, that is, the purpose of the statement is justify the need for the proposed research, not the review of literature. I know it is very difficult. That's what I said. The current state of knowledge relevant to the proposal. What has been done, what has not been done, and that's why you are using this one knowledge. Such things have been done. However, this particular work has not been done. We know that malaria is existing there, smallpox is existing there, or monkeypox recently existing there, but however, there is no treatment. That's why we're finding a vaccine for it. Oh, simple, over. Or SARS-CoV-2 infection is there, and we don't have any drugs. That's why we're finding a messenger RNA vaccine for this one. So that way you can make a statement. 5-line statement giving the what is existing, what is not existing, which you are going to fill the gap. That is the only few lines for writing the significance of result. The gaps that the project is intended to fill. There is a gap in the knowledge base or an unmet need that poses a problem. It persists. Some people say, oh, the existing drugs are all toxic one, produce a lot of severe side effects. So this plant extract is useful because it doesn't produce adverse effect. No, that is a silly way of making argument. How do you know the plant extract doesn't produce a side effect? Plants are always safe enough to be used. No, this is a vague statement. No plants are not. If you consume large amount, it also kills people. So that is again a bad thing. So you should not use the word saying that the existing English medicines are very bad. That's why plant extracts we are using it. It is safe enough to take. Again, that is one of the worst statement people make it. Every student copies the same statement. So you should not use that one. So for example, I've written here, the objective should be linked to this. For example, the development of a vaccine for SARS-CoV-2 or for malaria. As you can see the word. Not moving. Next slide. Hope it is not jammed. No. Okay. The rationale for the proposed research, the reason or basis for doing the study, ask these questions. Why is this experiment being conducted? Justify the need for a proposed research. What makes your research significant? Is the study to confirm, refute, or extend the previous findings, or to provide new findings or providing Yes, right. The present findings extend the previous ones, which have reported literature, and our study add more to the armamentarium of the existing drugs. For example, is the design primarily test research equipment? What are the practical implications and methodological advances of the study? Why this research topic important at this time? What innovations will come about? To conclude, you must demonstrate that your proposed study represents a significant step forward and that it will continue contribute to the literature and to the field as a whole. So you must come up with the reason why you do the work. I'll give some examples. For example, this is a simple statement to make it, not pages together writing. These novel compounds have the potential to enhance our armamentarium of heart failure therapeutics, and this gives a new mechanism of action. To say that one, or these studies will provide insight into the mechanism. This one, we hope that the findings will quell the debates over whether the drugs activate a key anti-aging protein. Such statement you can make it. An understanding of the digoxin's pharmacokinetics and a knowledge of the signs and symptoms of toxicity will enable the clinical pharmacist to play a pivotal role in the management of digoxin. This way he writes like this in the published paper. So this is a very nice statement they made it. These data demonstrate that azilsartan across the effective dose range has superior efficacy to the existing valsartan. Superior efficacy. It means dose will be 10 times less. The dose less means less adverse effects may be there. So this way these new findings are there. Enhanced adrenergic vascular reactivity. The results of the current study intend to provide information on the relative bioavailability of a compound X under fasted and fed conditions, which supports the ongoing clinical study. See, that's why we are doing this particular study, because it supports the study as it is our PK study shows. The methods described here are simple and can be successfully applied in quality control for precise and rapid spectrophotometric determination of rosuvastatin and ezetimimal combination drug products. So you made a good statement. People say, oh, yes, this study is important because it is giving something. In understanding, we show the first time how multi-layer polymer films, that somebody wrote it in the Journal Form Science. See, such things, when you read extensive literature, you should know that how people write it. Everybody has to make a statement. What is the novel findings of your project? So read those literature. From there, you copy that one statement, how they make it. Whether you can similarly you can write for your project. You're using their basic structure. On that basis, you can also construct sentences for your work. It's not copycat. It is only your formatting is looking at the format, how people write it. So you understand from the people, learners. So learn from the learners. The discovery of aliskiren, the first in a novel class of renin inhibitors that lowers blood pressure in several experimental and hypertension models, represents a novel potential treatment for hypertension and related cardiovascular diseases. So, in overall, what I described in all these things, so I represented by Venn diagram, shows the literature review. From that one, you get into topic and research question, which is the creative ideas, creative thinking, divergent thinking, convergent thinking. All these thinking paradigms comes into getting into this area, the violet one. From there, you develop into hypothesis. Then you get into aims and objectives one. Again, you apply creative thinking, critically analyzing, analytical thinking. Then you get into materials and methods, study design, statistics. Overall, what is the significance of the whole study represented this way? So putting everything together is an example of creativity. So what is creativity? The way you represent the whole thing into one picture, every day you will be using creative models. That is the definition of creativity. Myself coming down here and giving a lecture, putting everything from different sources is a creativity. You don't require any more examples. Saying review of literature, which of course Dr. Balkumar will be explaining in the next presentation. In summary, introduction section should effectively connect or weave the following sections: review of literature, research questions, hypothesis, aims and objectives, significance of the study, and the research. Use your experience in your experiment to achieve your expectation, that is research design and methods. The purpose is to describe how the research will be carried out. This includes plan of study, experiment design, statistical analysis, materials and methods, instrumentation, measuring techniques, data recording collection, difficulties and limitations, ethical and safety issues, timeline, work schedule, and experimental target. There are final research protocol. Of course, you may arrange this way or whatever your department or your university prescribes, but the more or less they come in the similar fashion, but with a different title they may give. But they should be in the same order. You cannot go in the reverse way, biography first, bibliography, then you cannot come down like this way. But structure remains same with a different nomenclature. So problem statement, which you're using it as a title, the introduction part, which we put all of them background, this is questions, hypothesis, and objective, significant study, everything comes under introduction. The same thing will carry forward in your published papers also, where you put everything in the introduction. About 4 paragraphs will be there approximately, of a page. Literature review, methodology, materials and methods, study design, that experimental study design. Based upon this design, you put the statistical analysis. Then work schedule, bibliography, references part. No, previous one, let's skip it. Write a quality protocol, that means present a clear and reasonable thesis idea. Relate the defined as a question to other relevant literature. Format hypothesis. Describe the method for investigating each objective. Based upon the hypothesis, you get into objective. Based upon objective, you write a method for each one of them. Conclusion, justify significance of the study, which is the significance of your research. Please move this one. Upon completion of the study, the research work should have made a worthwhile contribution to your field. This is worthwhile contribution. Here one ideas, remember. It proposes a new or improved concept, theory or model. It supports or disproves a concept, theory or model. It contributes new data. It offers new or improved solutions to analyze the data. It offers new analysis, procedures or new methods of analysis. It offers new ways to interpret the mechanism of action. In addition, the work should yield at least 1 or 2 publications in indexed. Keep in mind, which Dr. Balkumar also will be talking about it, indexed. That is Clarivate Analytic listed journals, Scopus index journals, not plagiarized, predatory journals where you pay 3,000 rupees per author, publish it in the predatory journals. That is not the publication, that is zero publication. There is no growth without learning, and learning is a continuous process. We always say that teachers are always a lifelong students. Similarly, learning is a continuous process. Let new beginning signify new chapter filled with pages of success and happiness. Faraday says, Michael Faraday, work, finish, and I added here publish also. Not only work and finish, but you must publish your work. Then it will be worthwhile that you stand in the in the line. Now I'll summary the what I said in the last 1 hour. The fundamental principles of scientific research are the formulation of a problem, the development of a study protocol, a research plan, and the description of the why, what is to be done, why you are doing it, and how are you doing it, what, why, and how. All 3 things should be described in your protocol. Keep in mind. About literature search and review, study design, statistical aspects, result analysis, and data interpretation. Our initial vision for a study largely shapes our actions, approaches, analysis methods, interpretation, and societal impact. That is the significance of study. Finally, ultimately, the study should be suitable for publication. The study, what you carried out, should be suitable publication and receive citations. Somebody should read our paper and cite it. Then only feel happy that yes, somebody is quoting me, indicating its quality and impact rather than being just a number. It is quality H index. People say, how many, what is your H index? 5, 10, 20, 30. So think always aim to have a good H index, more than 10. The scientific value or merit of published article is based on its own merits, not solely because it appears in a high impact journal. The article should be interesting, innovative, well designed, visible, and have the potential for impact, making an impact to the scientific community. Even more importantly, should be cited by other researchers. The scientific enterprise requires a lot of thinking and imagination before one puts ink on the paper. So this concludes my talk. Thank you so much. And see the 2 books. This is the 2010. We published the first time, From Idea. That is, we put as ideation to match this word publication. Such word was not existing in 2009, 2010. We came up with this word, again, creativity. Ideation, getting idea, which I discussed in the beginning itself, creativity, putting this one, and critical thinking gives idea. Ideation, then you finally end up in publication. That is the last part. So ideation to publication. And then followed with this 2010 book. Now this is the 2023 October we published it. Came out in the October. A copy of this one was presented to Dr. Ramesh. And last time when I came here, and of course it costs, whenever you get 50%, you can buy your own book because it price is $200. It is the previous book was very cheap. When it published was 500 rupees. It became 1,500. Now it is out of print. Nowhere you can get it, but still people are printing with black and white without no color diagrams. I removed it, but still they're selling for 1,500. Still it's cheaper one. But this book is expensive, but we have put it a lot of information here. And Dr. Senator will be giving a talk tomorrow morning. First talk will be his one on the clinical aspects. And Dr. Balkumar will be giving 2 presentations. After this one will be next one. That's why we put this one, how we are moving from here, imagination to target discovery, then to preclinical to clinical to approved new molecule, and then publishing and grantsmanship. See how we move here. So this is quintessence of basic and clinical research. And finally, everybody has to do publishing, whether you do basic research or a clinical research, but scientific publishing is a must. Thank you so much. You can ask whether breaking for a lunch.
Thank you so much, sir. Both Dr. Jagdish and Srinivas Raju. I think, sorry for keeping you a little wait, but I think nothing will come easy. So I think once again, as a token of our love to the both people, right? Since Jagdish will be available tomorrow, now I would like to honor Dr. Srinivas Raju with a small memento. So give you a big round of applause. And he is instrumental in connecting me to Jagdish. So I want both of you to be on the stage, sir, please.
Please give them a big round of applause. So your presentation was a dose of knowledge, extraction of complexities of acute toxicity testing, and a catalyst for new ideas and perspectives on dose selection that the audience sorely needed. Thank you so much, sir. We are honored to have you had us share your knowledge with us. Yeah, I'll announce the lunch. Okay. So one more.
Yeah, because so that it will be remembered for him, right? Yeah.
Thank you so much, sir. We have reached the midpoint of our program, so let's take a quick 40-minute break, lunch break to recharge and back refresh. Come back to the auditorium by 1:40. Thank you.
Post lunch, we'll be having the session of Balakumar, Dr. Balakumar. So AI on literature review.